Thank you very much for inviting me to this exciting conference at Tijuana Innovadora. My name is Ron Diskin. I'm a scientist at the Weizmann Institute of Science in Israel, and I'm going to present you today the power of structural biology in fighting viral diseases. So I hope you can now see my screen. And before I start, I want to explain a little bit some terms in my title. So the title is Structural Biology in the Fight Against Viruses, but what structural biology really is all about. So structural biology is actually a discipline that aims to understand the biological systems using information about their spatial organization. And by spatial organization, you understand that we want to see the biological systems that we investigate. And when we investigate proteins, we really want to see how they are organized in three-dimensional. And for that, we use fancy instrumentations like this synchrotron radiation facility that you can see here that is located in Grenoble, France, or electron microscopes, state-of-the-art electron microscopes. And eventually, using those fancy techniques, we can draw a three-dimensional comprehensive map of proteins. And these maps show where are the exact locations of all the thousands of atoms that make these proteins. And we can represent that in neat graphical ways, as you can see here. And why do we even care to see the structure of these proteins? We do that because the function of the proteins relate to their structure. And if we have structural information, we can derive functional information. If we understand the function of the proteins, this is the starting point to manipulate the function. So for example, to inhibit proteins or to change their specificity. And this is basically how we today design novel drugs against some of the interesting biological targets. Now, the other term that I want to pay attention to is viruses. What viruses are? So viruses are tiny particles that basically do not have their own capacity to replicate. And that means that in order for a virus to replicate and disseminate itself, he really needs to find a host cell. This host cell could belong to humans, it could belong to animals, even plants. But the virus really needs to find a way to penetrate this host cell in order to replicate. And the essence for that is the lack of some components that viruses simply don't have, like ribosomes, for example. So this very intricate process of viruses that need to locate their cells doesn't really happen in vacuum. Typically, there is some kind of an immune system that is searching for such viruses and trying to prevent them from entering the cells. And this is exactly what we do in our lab. We study how viruses recognize host cells, how viruses penetrate host cells, and how they do that in the face of the immune system, and what are the tricks that they are sometimes using in order to evade immune response, and how we can actually use this information in order to combat viruses. So the outline of my COVID-19 talk today would be a short overview about coronaviruses. I will talk a little bit about the drugs and vaccine efforts that are being developed and pursued nowadays. And I will also tell you about the things that we do in the lab, which is targeted immunotherapy against COVID-19. And the goal in this talk would be to give you a perspective of a scientist, how we actually see this pandemic, some things that the information that we have in our hand, and I hope it will be interesting to all of you. So coronaviruses, or coronaviridae, is a large family of viruses. They're enveloped, meaning that they have a membrane coat around them that surrounds the genetic material. And you can see it very nicely here in this micrograph. You can see this round shape of the viruses that is caused by this membrane coat. They also have a single-stranded RNA genome, and that will become important later on in my talk. So their genome is coming in the flavor of an RNA molecule. Now you can see here in this very nice micrograph that was taken many years ago, density around the viruses. This density belongs to the spikes. Those are large protein complexes. And this unique shape gave them the name corona because it looks like a crown. And here you can see that we have many viruses. This is actually a very partial phylogenetic tree showing some of the viruses that we know, and this is the new virus that emerged. So what is the history of coronaviruses? Actually, it goes back quite a long time. The first virus was actually identified in the 60s. That was a virus that infects chickens. And even nowadays, this is an agriculture problem. But that was not the only virus that we got familiar with from this family. We know about other coronaviruses that actually infect humans. And those are very common viruses. They are very widespread. Many people contract these viruses every year. And they typically only cause mild common cold symptoms, sometimes pneumonia, and no more than that. Now, the first instance of a real dangerous coronavirus was the emergence of SARS, or Severe Acute Respiratory Syndrome virus. And that was in November 2002 in China. And this virus infected almost 10,000 people. It killed almost 1,000 people with a death rate of about 10%. And then in July 2003, it just went away. It disappeared. So that was the first encounter with the deadly virus from this family. Now, the second encounter was in 2012. Another virus emerged. This time, its name was the Middle East Respiratory Syndrome because it appeared in Saudi Arabia. And you can see that this virus did not affect as many people as the first virus because the spread of the virus is much slower. However, it's more deadly. Almost 1/3 of the people that contract this virus die. And this virus is still spreading, and you can see that every year we have additional few cases of this virus. So it's still out there, but it's not causing a massive pandemic because it's spreading very slow. And of course, our very recent encounter with the coronavirus, that was late December 2019, again in China, in Hubei province. It's called SARS coronavirus 2 because it's very similar to the first coronavirus that appeared almost 20 years ago. And it's causing the coronavirus disease, or COVID-19. And this is how we currently experience this virus, social distancing and all kinds of different measures trying to restrict the spread of the virus. Now, this slide showing you the epidemiological data that is accumulating in the world, and I took that from the ECDC. And basically, you can see the number of confirmed cases every day since the beginning of this pandemic. And I want to show you basically the magnitude of the spread of the virus. Here, I don't know if you can see that, but this very small blip that occurred in February this year was the massive outbreak in China that all the world watched very carefully. And we were all fearing that it will eventually spread to the rest of the world, which eventually happened. And you can see how large the spread of the virus is nowadays. In all the different continents, we see the virus, and there is massive infections going on, which really, the beginning of the infection in China looks very, very small compared to the numbers that we're seeing nowadays. So how can we really stop the SARS coronavirus? So basically, we need to consider two arms or two different tools that we have for prevention and for treating people that already contracted the virus. Now, for prevention, besides social distancing that is very helpful, the only medical instrument that we have is vaccine or vaccines in general. And I will talk a little bit about the vaccines. Now, once people already contracted the virus, then we're talking about the treatment options, and there are various treatment options that I will go over in a minute. I just want to mention that there is another possibility. It's called prophylactic treatment. And basically, that combines those two arms. And the idea is that if we have an effective medicine against the virus, we can actually give it to people before they contract the virus and protect them. Now, of course, this is not something that you can do massively to a large number of people, but you can certainly do that to a few people that you want to protect, like healthcare workers, for example, or other people that we want to protect. And that might be achievable by some of the compounds or therapies that I will mention today. So I want to start with off-the-shelf drugs. Drugs that are already available on the shelf, and we can now use them in order to fight the virus. And that immediately raises the question of how could it be that we already have drugs against a newly emerged virus. And people actually asked me this question before, and if we don't know what is the answer for this question, that of course could lead to all kinds of different theories for the virus. So I will explain how come we can actually have good drugs against the virus, although we only now encounter this particular virus. In order to understand that, we actually need to look at the drugs and divide them into two parts. There are drugs that act on the host on one side, and there are drugs that act on the virus on the other side. So let's start with the simple case of drugs that act on the host. And the very simplest thing to illustrate that would be ibuprofen. If you have fever, you can take some anti-fever medication or anti-pain medication. And this is regardless to the cause of the fever. And of course, it will be also effective in reducing fever for COVID-19 as well. But not only that, we have other drugs that are more targeted for the virus, and or at least for the COVID-19. And one of the drugs that made it to the news is dexamethasone that you can see here. This is a steroid. And in order to understand how this simple steroid that is very common, very cheap, widely used for many decades, how this could be effective against the virus, we need to appreciate the nature of the disease. So the more severe cases of COVID-19, the ones that people are eventually getting hospitalized and make it to the intensive care units, they don't really suffer from the virus directly. They actually suffer from a condition known as a cytokine storm, which is an inflammatory condition that basically results from overactivation of the immune response. And the immune response and the inflammation starts to hurt healthy tissues in vital organs. And that could be, of course, a deadly thing. So steroids in general reduce the immune response. So by giving those steroids, we can reduce the inflammation, and that can help to save lives of severely affected patients. So that helps to fight COVID-19, but not directly fighting the virus. But we also have drugs that are useful against the virus. In order to understand how these work, we need some additional information. So here you can see a schematic diagram of the genome of the virus, which I already mentioned is an RNA virus, meaning that it's coming in the flavor of an RNA molecule. Now here in boxes, you can see the different proteins that are encoded by the genome of the virus, and there are several of these proteins here. And I will circle some of them, because those are drug targets that we currently pursue. I will mention S later on, and also the PL and the 3CL proteases. But now I want to focus on another protein that is called RDRP, or RNA-dependent RNA polymerase. Now you need to understand that each and every RNA virus, and there are many RNA viruses out there, need to bring with him an RNA-dependent RNA polymerase in order to replicate. If it doesn't have this capacity, it won't be able to replicate. So this is a very important protein that all RNA viruses have. Now, if we look at structures of RNA-dependent RNA polymerases from various viruses, starting from dengue here on the right, poliovirus here in the middle, and the original SARS coronavirus from the early 2000s here on the left, you don't need to be an expert structural biologist to understand immediately that we're talking about proteins that are very similar to each other. They have the same features that repeat in all the different cases. And this is not very surprising because these are proteins that really need to carry the same function, which is to read an RNA template and synthesize a new complementary RNA chain. So it's not wonder that we have such a big similarity between those proteins, even though they're coming from very, very different viruses that don't even belong to the same family of viruses. So you can also now appreciate that if we can inhibit one of these proteins using a drug, the drug could potentially also be effective against other viruses by inhibiting their RNA-dependent RNA polymerases. So immediately when this virus, the current SARS coronavirus emerged, people started to screen known drugs to see which one would be beneficial and will be able to stop the new virus. And this is one of the early works that published early this year, and you can see a series of compounds that were tested against the new virus. And here in this box, you can see one compound that you may have heard its name. This is remdesivir. And first of all, you can see that it inhibits the virus. You can see that in this red curve. And also that it's not highly toxic to the cells, which is a good property of any drug that we want to use. So this is an inhibitor of the RNA-dependent RNA polymerase. And it belongs to a family of inhibitors that you can see here that are called nucleoside analogs. Basically, they resemble to the building blocks of the RNA, and by this similarity, they can bind the RNA-dependent RNA polymerase and inhibit its function. And remdesivir is a molecule that was developed by Gilead Sciences, and it was originally developed to fight Ebola. However, it turned out to be that it's not the best drug to inhibit Ebola, so it was left on the shelf. And now, in the face of the new pandemic, it was tested against the new virus, and you can see that it indeed can inhibit the virus. It went into clinical trials, and it was shown already that there is some benefit in using remdesivir. So now people are actually getting remdesivir in order to alleviate the symptoms of COVID-19. And you can see that there are other drugs that are nucleoside analogs. They can also inhibit the RNA-dependent RNA polymerases. This molecule, for example, was developed against flu, but it's also has some partial activity against SARS. So there are additional drugs like that, and some are still being tested in the clinic, and so on. So I showed you how we can actually have some already made medications that could potentially be effective against the new virus. Now I want to jump and talk a little bit about vaccines. Now I'm bringing this table to show you many different efforts to develop vaccines against SARS. But if you will pay attention to the citation here, you will see that this article is from slightly over 10 years ago. So this is not the current SARS, this is the previous SARS. And you can see that there were many, many different studies trying to develop vaccines against the old virus. And I'm showing that to you just to illustrate that there is more than one way to achieve a good, viable vaccine against the virus. Now, most of these approaches make use of a very simple strategy. And the idea is to present a key protein, a key viral protein to the immune system, to teach the immune system how this viral protein looks like, to train it, if you like. And then once the real virus comes, the immune system is already recognizing this virus as a foreign agent, and it can readily mount a good response. So the protein that most of these studies have used is this beautiful spike protein that appears here around the virus. So those are the white densities that you see here are made by this beautiful spike complex that its structure was solved a few months ago, and you can see here the citation. And most of the efforts are actually to take this protein in one form or another and present it to the immune system. So one of the leading companies now in the race to develop a good vaccine is Moderna. I'm sure you heard about this company. And let's try to understand what Moderna is doing. So Moderna is not taking this protein per se, is actually taking a messenger RNA molecule that encodes to this protein. So they're taking this messenger RNA, they inject a large amount of this messenger RNA into the subjects. Some of the cells of the healthy tissues of these subjects take this RNA molecule and they start to produce the protein. And once they produce the protein, then the protein is seen by the immune system and antibodies are being raised against the protein. Now, conceptually, this is something that exists in the research world for a long time, but we don't have an approved vaccine that is using this technology so far. So if the vaccine of Moderna will be approved, that will be the first time that an RNA-based vaccine will be approved. But there are many other ways to present the spike complex to the immune system, and I want to show you another example. And this is the use of some viral platform to present the spike complex to the immune system. So here in the left, you can see an example. This is a virus that is called VSV. It normally doesn't really infect humans, and when it does infect humans, it causes a mild disease. But we can engineer this virus now to present, instead of its own spike complex that is shown here with these blue marks, we can engineer it to display a different spike complex. And in this case, it was engineered to display the spike complex of Ebola virus. And that created this recombinant virus that is called RVSV-ZIBOV, which is basically the first vaccine against Ebola virus that was approved. It's called, the commercial name is Ervebo. It's being produced by Merck, and it was approved as the first vaccine against Ebola only this year. So this is a very new technology. And I should mention that the VSV is only one viral platform. There are actually several good viruses that we can utilize, and some of the leading companies now in the race to develop vaccines against SARS coronavirus are actually making use of this platform. They're using viral platforms to present the spike complex of the virus in order to elicit good immune response. Now, we know that this way of presenting the spike complex is good because we actually investigated that. So we took this RVSV-ZIBOV and we analyzed the ability of this vaccine to elicit an immune response in vaccinees, and we compared that to the immune response that survivors have in their blood. And this is some early work that we conducted in the last couple of years. And here you can see one slide that summarizes the work. And basically, we in collaboration with the group of Florian Klein in Germany, in Cologne, Germany. We took antibodies from people that were vaccinated with this vaccine. We analyzed the antibodies. We deciphered how these antibodies target the spike complex of Ebola. In this case, you can see here the spike complex of Ebola. And we saw that the way the spike is presented to the immune system can actually produce very good immune response, and we can target very beneficial epitopes, targets on the spike that eventually allow the immune system to mount a very good response that is effective. It can really neutralize the virus. So this is a very potential way to move forward with the vaccines. And as I told you, there are many, many such efforts, and they vary really by the vector that they're using. So we talked a little bit about off-the-shelf drugs and vaccines. Now I want to go back and talk about specific drugs that are being developed against the virus itself. And one of the major targets for such drugs is the protease, or are the protease, because we have a couple of proteases here. We have a couple of proteases here. They're shown here. And the proteases are special proteins that basically designed to cleave the polypeptide chain that is the precursor of the virus. And you can see the virus cleaving points here with the arrows and the triangles on this polypeptide chain. And without this cleavage, basically no infectious particles will be produced. So if we can inhibit the activity of these proteases, we can actually prevent the virus from replicating. And this is a structure of one of the proteases that was already solved and is available already in the databases. And it looks very, very, very similar to the proteases of the previous SARS that emerged in the early 2000s. And this is not very surprising because the viruses are really, really close to each other. And they're basically identical in the site, in their active site, which is the target of many of the inhibitors of these proteins. So there was a large body of work that was already conducted with the previous virus, and there were many compounds that were found to inhibit these proteases. All of those compounds are putative drug candidates. None of them made it to the clinic because the previous SARS really disappeared after less than a year. But all of these are good, at least starting points to develop drugs. And on top of that, there are massive efforts nowadays to develop new drugs. And I want to mention only one such effort, which is the COVID Moonshot Initiative, which is really a crowdsourcing initiative to recruit experts from all over the world to design novel drugs against this drug target. And they already have some compounds that looks very promising, and it could be that some of them will progress into clinical trials in the future. So another potential way to fight the virus is using immunotherapy. And this is a very promising approach because we know from a large body of work that was conducted in recent years that immunotherapy is effective against various viral diseases. And there is a great hope that it will be very effective against COVID-19 as well. Now, the very crude way of using immunotherapy is to take plasma from people that recovered already from COVID-19. Those people already mounted a good immune response that allowed them to recover, and their plasma is rich with antibodies that can target the virus. And now you can give these plasma samples or plasma donations to critically ill patients, and that really alleviates their symptoms and help them to recover. So this is a good way that I think most of the countries today utilize, and it seems to be effective, although there are still clinical trials that are trying to evaluate the amount of how much these plasma samples are actually effective in fighting the virus. But this is not the only way, and now I'm going to show you some of our own data that relate to immunotherapy that we actually develop in the lab. We are not relying on plasma samples from donors. And before I will show you the results, I just want to mention that research in general is a collaborative effort. It takes many people, intense effort, and a lot of funding in order to conduct the research. And I want to acknowledge all the people that are involved and make everything possible. So starting with my lab members and former lab members, all of our excellent collaborators that make it possible to conduct this research, and the many funding agencies and donors that really contribute funds for this research. So our idea is to develop antibody-like molecules that are known also as immunoadhesions. Now, the idea of immunoadhesions is very old. There are several decades have gone since the first immunoadhesion was conceived. And all the immunoadhesions has this very generic form. They look like antibodies. They have two active domains that are aimed to bind the target in some way or another. Sorry. And they also have an Fc portion that is borrowed from an antibody that helps these molecules to recruit the immune system, basically. So we already designed immunoadhesions in our lab, and I'm showing you an example for one immunoadhesion that is called Arenacept, that is targeting different viruses that belong to the Arenaviridae family. It can target several different viruses, and we show it here. And those are highly deadly viruses. You can see that in order to work with these viruses, you really need to work in the highest safety lab measures that could be taken in a BSL-4 facilities. And you can see that it's very effective in neutralizing various viruses. And we were wondering if we can design something similar also to combat SARS. Now, in order to combat the virus, we need to have an active domain that would bind the virus. And we actually taking advantage of the natural life cycle of the viruses, which is they need to enter the cells, as I mentioned previously. And in order to do that, they need to locate the host cell. And they do that by their spike complex that we mentioned already. And this spike complex binds a specific receptor on the surface of cells. And we can use this cell surface receptor as the active domain that would eventually bind the spike complex, and basically we'll have a decoy. And if we will saturate the spike complex with our decoy receptor molecule, it won't be able to locate the target cell, it won't be able to enter the cell, and then will prevent the replication of the virus. Now, we already know what is the receptor of the virus. It's a molecule called ACE2. And this is a beautiful structure that was presented earlier this year. And here you can see the viral protein here in blue. And you can see that it binds the cellular receptor here in magenta. And the idea would be to take this part of the receptor, hook that into the Fc portion of an antibody, and create the immune adhesion. Now, another subtlety that we need to consider is that this virus is a zoonotic virus. It's a virus that emerged from animals, crossed from animals to humans. And evolutionary, this virus is actually optimized to infect its host, natural host, which could be bats or pangolins or any other animal that we failed to recognize so far. So it's not optimized to humans at this point. Maybe in the future, if it will circulate in the human population for many years, it will become optimized for humans. But now it's still optimized to its natural host. So if we will take ACE2 not from humans, but from its natural host, that might give us a much better immune adhesion against the virus. And this is exactly what we were trying to do. The only real problem is that we don't know what is the host that really transmitted the virus to humans. And here we see the power of structural biology. So on the left, you can see the structure of the viral protein in gray, and you can see the cellular receptor in orange and yellow. And the key contact residues, which are the building blocks of the protein, you can see here in this stick representation. And now we can look at the sequences of these ACE2 receptors from all the mammals. We just take database of sequences of mammals, and we look at the sequence diversity of the ACE2. And here with these arrows, I'm indicating sites that are critical for the binding of the virus. And in each of these sites, you can see that we actually have many different letters. These are one-letter codes of amino acids. And having different letters here tells us there could be different amino acids that appear in different ACE2 in different mammals. So what we did was to take this information, this structural information, and simulate, using a computer, simulate what would be the binding of various ACE2 coming from various mammals to the virus. And here you can see some metrics that we use in order to evaluate the interface between the viral protein and the human and the ACE2 receptor. And you can see here each dot represents a different ACE2. You can see the human in green and all the other mammalian ACE2 as different spheres. And then we can isolate and look at the ones that really represent the top binders, the ones that can bind very tightly to the viral protein according to the prediction here, and see what are the structural features that allow them to bind so well. And then we can take these structural features and embed them into an ACE2 that was coming from humans. And this is exactly what we did. And you can see here how it actually looks like. So in orange, we have the original ACE2 from human. And in blue, you can see a modified version of ACE2 in which we modified one of the building blocks in order to enhance the interaction with the viral protein shown here. So in this example, we have a longer side chain that makes better packing with these residues of the viral protein. Here you can see that by changing the amino acid with a slightly longer neck, we can achieve a salt bridge that will form with the viral protein. Again, if we slightly modify some of the residues, we can better organize their surface and create an additional hydrogen bond with the viral protein. So by combining some of these modifications, we were able actually to produce a superior ACE2 that would bind much stronger to the viral protein. And here you can see the actual experimental results. So this is a binding curve. And to evaluate binding, we can actually look at a number that tells us about the dissociation of the two proteins. So this is the dissociation constant. And as lower this value gets, the tighter the binding is. And this is the binding of the wild type or the human ACE2 receptor to the viral protein. And here is the binding of our modified receptor to the viral protein. And you can see much tighter binding. You can see that this value now is much lower. So we achieve much tighter binding, which is always good for us because tighter binding means that the reagent will be able to act at lower concentrations in a better way against the virus. And we can actually test the biological action of this reagent against the virus. And you can see here a slide that summarizes the biological results. So you can see that we can neutralize pseudoviruses. And in gray, you can see an immune adhesion that is made from the human receptor compared to the red curve, which is the modified receptor. And you can see that there is a big shift that tells us for more beneficial neutralization. Here you can see the ability of this reagent to identify infected cells, which is critical if we want to recruit the immune system to fight and kill infected cells. And you can see that it can do that much more efficiency compared to the much higher efficiency compared to the human reagent. And here you can see the neutralization of live viruses that was tested by collaborators in proper biosafety measurements. And you can see a huge difference in the ability of our modified reagent to neutralize the virus versus the human reagent. So basically, we have a very, very potent reagent that is a candidate for clinical use. So this is one way to produce immunotherapeutic reagents, but of course, there are additional ways. And one of the ways that is very heavily explored all over the world is to isolate monoclonal antibodies from infected individuals. So I showed you before that we can take plasma samples from those patients. But the plasma sample contains all kinds of different antibodies. And of course, we want the ones that are highly reactive against the virus, and we cannot isolate them from the plasma samples. But we can identify such antibodies. And if we manage to identify such antibodies, we can produce them. And then we no longer need donations of plasma from patients. We can actually produce them recombinantly in the industry and use that as drugs against the virus. So there is massive effort all around the world. Some of the companies like Eli Lilly and Regeneron already initiated clinical trials with their own monoclonal antibodies that they've isolated from patients. And there are additional many companies and labs all over the world that are trying to do exactly that. And there is room for many more antibodies because eventually we will want many companies that will produce good antibodies, and we want large diversity of antibodies also. And we were also engaged with such an effort. And again, this is in collaboration with the lab of Florian Klein from Germany. What Florian did is to take samples from people that recovered from COVID-19, and he took samples along a few weeks after the recovery, and he looked for the antibody and the way the antibodies develop along this time. And he isolated quite a bit of monoclonal antibodies from these patients. And some of them are really highly reactive against the virus, and you can see them here. Some of them are already given to an industrial partner for further development. And now we want to investigate some of these antibodies with our structural abilities, because we want to understand how these antibodies actually exert their function on the virus. We want to understand how they bind the virus, how they neutralize the virus. And having this knowledge will really help us to advance some of these antibodies into clinical use. So I'm going to end my lecture with one slide that I will share with you, with the most recent structural information that we derived in the lab. And here you can see an antibody or the Fab portion of an antibody on the right, and on the left you can see the target viral protein. This is the receptor binding domain of the virus, shown here with this light blue. And from this structure, we can learn exactly how the antibody binds the viral protein. We see exactly the binding interface. We see exactly what are the important amino acids, what is the epitope of that the antibody recognizes. We can also learn immediately what is the neutralization mechanism of this antibody that basically competes with the binding of the receptor, the ACE2 receptor. So we have the mechanism and we have the epitope, and we have a lot of vital information that will help us to advance these proteins. So this is what we do in order to fight the virus. And I thank you all for listening to my talk, and I'll be more than happy to answer questions in case you have any. And thank you very much.
Bravo, bravo.
Thank you.
So it was great to hear your talk, and what I really like it's that it's made for people that really don't know anything about biology and chemistry, and so thank you very much. I have a couple questions, but I would just, because the time, I'm going to choose one. Okay. And it says, what makes a virus more or less difficult to find a vaccine?
That's an excellent question. And we have a long history of viruses that resist efforts to develop vaccines, like HIV, for example, which for many, many years people have trying to develop vaccines, and so far we still don't have a good vaccine against HIV. And also influenza. We also know that each year we need to get vaccinated again, because the vaccine of the previous year is not effective against the viruses that keep appear every year. I don't think SARS coronavirus is one of these examples. We know exactly why we can't develop good vaccines against influenza, because it's a virus that keep changing its genome. It recombines different parts. This is because of a very special structure that its genome has. We also know for HIV some of the difficulties or some of the aspects of HIV that make it extremely difficult to develop vaccines. Luckily, we don't see these features in SARS coronavirus. So SARS coronavirus is actually more similar to viruses like Ebola, for example, for which we actually have good vaccine. So I'm very optimistic, and I think we will have good vaccines against SARS coronavirus, and I think we will have more than a single vaccine, which is great. I don't know what would be the efficacy, what would be the protection level that these vaccines will offer, but any kind of protection would be great, and it could really substantially influence the course of the pandemic.
Okay, great. Thank you very much. Thank you for being here, Dr. Ron Diskin, and we really appreciate it. And please be safe. Thank you for being with us.
Thank you very much. Thank you.